Once you understand how DNA is used to make a messenger RNA and how the messenger RNA is translated into a protein, you can understand how mutations in DNA will cause an effect on a protein. Here's a cell with a nucleus, a strand of DNA with a whole lot of base pairs. We're going to be interested in just one triplet, an ACC, which matches a TGG. As the protein moves along and translates a messenger RNA, we'll have a single strand with bases. Our TGG will be a UGG. So copying from the A gets a U, uracil, instead of a T. C matches up with the G, and C with a G. That strand will move out through a ribosome. When the UGG is in the ribosome, that will pair up with an ACC. That will be attached to an amino acid. That one happens to be a TR. The result will be a growing amino acid chain in which there's a TR, and remember the TR is wider at one end than the other, and so it'll make a bend as our amino acid chain keeps growing. If that's a protein that connects two cells to each other, then that bend as a part of the protein, it will be a part of what makes that protein hold on to the membrane, maybe attach to parts inside the cell and attach to the protein on a neighboring cell. That's important for your skin to hold together, for your organs to hold together. Anytime cells need to be attached to each other, these kinds of attachment proteins are critical. So that's what normally would happen. Now let's look at what happens if we have a mutation. Either radiation or some chemical came in and cut our DNA and removed that AT. And that when it was repaired, instead of an AT, by mistake, there was a GC in place. Now, instead of an A pairing up with a U to make a uracil, we're no longer going to have that U. Instead, we're going to have a C, a CGG on our messenger RNA. When we get down to the ribosome, we've replaced that U with a C, and as the messenger RNA goes through the ribosome, we now have a CGG, which will pair up with a GCC instead of what it would have been an ACC previously. That means that we aren't going to have a TR here. Instead, we're going to have an AR. And an AR is a long, thin amino acid instead of wider at one end than the other. In our mutated protein, we're going to put an AR instead of a TR. Now we've lost the bend that was there before. This protein is now a different shape. Before it had a bend that might have mattered for its structure, it's now a little bit different. What if that bend makes these proteins attach less strongly? It makes the attachment weaker. What that means, every time something pulls on the skin, as the pull gets stronger and stronger, it's more likely to break that attachment, and these cells will disattach. And that might cause easy bruising. But maybe it's the opposite. Maybe instead that change makes the attachment stronger. In that case, we might actually have an advantage. The skin or the organ will be less easy to tear, so maybe there'd be less bruising, but there might also be harder skin or organs, less flexibility. Both flexibility and bruising and tearing matter. If skin is too hard or organs are too hard, they can't flex and move as you do. If they're too loose, bruising is easier. Different mutants might make it stronger or weaker. The two different possibilities, the original protein and the mutant, each has potential advantages and disadvantages. The one that makes it weaker might have more flexible joints. It might be easier to bend into positions to do something. There are known mutations of these proteins that connect cells that increase flexibility, which people are often happy to have, but also may make bruising or damage a little easier, which people are unhappy to have. And there are differences that might make the attachment stronger, which might make stronger connections and less easy to tear, but potentially harder skin or joints that don't bend as easily and less flexibility. So the mutation, that little change back here of a TA to a GC, led to a change in messenger RNA, changed what would have been a TR to an AR instead of a bend. We had a straight part of the protein, and that made that protein attach more strongly or more weakly. That mutation affects the protein, which affects phenotype, how the organism functions.